Hello my dear students, welcome to our channel Rajesh Biology and I am Rajesh MSc Zoology. Today our topic is oogenesis. We already completed spermatogenesis process. Spermatogenesis means the formation of the male gametes in the testes. That is, we already completed in the previous class. Now we are going to discuss about the oogenesis, formation of the female gamete in the ovary. And it is completely different from the spermatogenesis process. How it is so different from the spermatogenesis? Spermatogenesis process begins at the age of puberty. But oogenesis process, it begins at the fetal stage and it stops also at the time of a fetal stage. So it is better to divide the oogenesis process into two steps, before the birth and after the birth. Okay, so I'll do one thing. I'll divide this oogenesis process into three stages, not two, three stages. That is prenatal development, that means within the mother's womb, when a female baby is present in the mother's womb, prenatal or before birth, and after birth up to the puberty, after birth or birth to puberty, and from the puberty, every regular menstrual cycle. Okay, so at the age of puberty. So like this, we can divide the oogenesis process into three stages. First, we will discuss about the what happened in the prenatal female baby. That means when one woman is present in her mother's womb, in her ovary, how the oogenesis process takes place. Just imagine as it is the baby's ovary. It is what? The ovary of the prenatal baby. So it is the baby which is present in the mother's womb. Placenta. So it is the ovary of the baby when the baby is present in the mother's womb. In this fetal ovary, the primordial germ cells are present. The primordial germ cells are going to undergo mitotic divisions and they forms the oogonial cells. Just like the germ cells produce spermatogonia in the spermatogenesis process. As like that, here the primordial germ cells are undergo mitotic division to form what? The oogonial cells. And these oogonial cells again undergo mitotic division, number of mitotic divisions. I'll do one thing, I'll erase this. I'll draw a big ovary, female fetal ovary, or I'll draw here. It is the female fetal ovary. It is what? A female fetal ovary. This is the primordial germ cell. The purple color structure is what? Primordial germ cell. This primordial germ cell undergo mitotic division to form number of oogonia. Okay, these all are oogonia, and a few oogonia are covered by some follicular cells. These cells are secreted by the ovarian wall. The ovarian wall is going to secrete some cells. That cells are surrounding these oogonial cells. Entire development, what we are discussing now, it is happened in the fetal ovary. So these are all oogonia, and it is the primordial cells. All these cells covers the oogonial cells. And in this course, in this development, the oogonial cells undergo mitotic division and they increase in their number, and nearly a couple of million oogonial cells are formed within each fetal ovary. So 1 million is present. For example, 1 million oogonial cells are present in one ovary. Another 1 million oogonial cells are present in the another fetal ovary. Once they reach their maximum limit of number, they stop their mitotic divisions. They stop their mitotic divisions. After stopping their mitotic divisions, one of the oogonia increase in size by suppressing the other oogonial cell growth. One of the oogonia increase in size. So the remaining are going to degenerate. This is the oogonia, and these are the primordial cells which are present surrounding this oogonia. Once only one oogonia is there, that oogonia is differentiated into primary oocyte, same as like in the spermatogenesis process. The spermatogonium is developed into, differentiated into the primary spermatocyte. As like that, this primary oogonia, a primary oocyte is formed within the follicular cells. So now it is called what? Primary oocyte. Once the primary oocyte is formed, there is no mitotic divisions. Primary oocyte is undergo only the meiotic division. In spermatogenesis also, primary spermatocyte undergo meiotic division, first meiotic division. Here also primary oocyte undergo what? First meiotic division. It starts, it's a first meiotic division. Okay. So when the primary oocyte is there, this follicular cells are arranged around this. These follicular cells are just like a simple epithelium. It is look like a flat epithelial cells. The flat epithelial cells are called what? It is called primordial follicle. The primordial follicle is present. Within the primordial follicle, there is a oogonium that is differentiated into primary oocyte. Primary oocyte is now ready to undergo meiotic division, and it starts its meiotic division. Once it starts its meiotic division, it gets arrested in the first meiotic phase. That means prophase 1. In the meiosis 1, in prophase 1, in the diplotin stage, in early meiotic division, at the diplotin stage, it is going to arrested, and it stops its further division. It get arrested. Okay, so it is the primary oocyte which is arrested at the prophase 1 of meiosis 1, and these follicular cells are present. Primordial follicle, now it is called what? Primary follicle. Primary follicle contains primary oocyte that is arrested in the meiosis 1 and prophase 1 at diplotin stage. This type of arrested cells are present in the fetal ovary. How many? A couple of primary oocytes are there. When all are primary oocytes, all the oogonia are converted into primary oocyte, there is no further development, further formation of oogonia. The primary oocytes which are present in the fetal ovary, they are the final. There is no other oogonia, there is no other formation. But in spermatogenesis process, the male germ cells, the spermatogonial cells, are going to produce the sperm cells up to the death of that person. But in this condition, the oogonial cells, once they are converted into the primary oocyte, there is no further oogonia. This is the development of oogonial cells into the primary oocytes in the female fetal ovary. Is this clear up to now? Now we will going to discuss about the puberty. At the age of puberty, what will happen? At the age of puberty, these primary oocytes which are present in the primary follicle, they are going to degenerate. They are going to degenerate. The number of oogonial cells, the number of primary oocyte cells are going to decrease in number. How much they decrease? At the fetal stage, there is 1 million, and at the time of the birth, they are 1 million, and gradually when the baby is going to develop into the adult up to the puberty, the number of primary oocytes are going to decrease step by step, and they reach 60,000 to 80,000, 60,000 to 80,000. Like that, they degenerate in number. This is what happened in the aging, in the process of aging. Once one female reaches the puberty, These primary oocytes, they are released from that stage, they continue their division. That is we are going to discuss now in the puberty. Okay, at the age of puberty, in which stage the primary oocytes are arrested? The primary oocytes are arrested in the meiosis 1, prophase 1, diplotin stage.